Bismillahirrahmanirrahim. I am Sayyid Mudassir Ali Mashadi from Allama Iqbal Open University, and I am feeling very privileged to have a talk at the first international conference at University of Bukhara. The topic of my talk is pharmaceutical co-crystallization as an emerging trend to modify the physicochemical properties. These are the outlines of my today's talk. First of all, I will describe some introduction and history of pharmaceutical co-crystals, and then I will describe how we synthesize our co-crystals and characterize them with different techniques like IRPXRD, single crystal XRD studies, and solid state NMR. And I will have a special emphasis on solid state NMR because it reveals the different aspects, and then at last, I will describe the physicochemical properties of the pharmaceutical co-crystals which we synthesized and antioxidant studies. The dawn of this field dates back to the Nobel Prize in Chemistry 1987, which was awarded to Jaquem, Lehen, and Pedersen because they devised the supramolecular chemistry and crystal engineering. The supramolecular chemistry is the chemistry beyond the molecule, which leads to the crystal engineering, which is understanding of intermolecular interactions in the context of crystal packing and utilization of such understanding in design new solid with desired physical and chemical properties. And especially this can be applied to the drugs or pharmaceutical science. Now, the important question, what the pharmaceutical co-crystal is. The co-crystal is a multi-component crystal in which all its components are neutral and solid under ambient conditions when in pure form. Means the two different compounds which are neutral and they are combined with each other without having any ionic or covalent bonding. Like in this case, you can see the two compounds which are joined by the hydrogen bondings. And these co-crystals have very important impacts on biomedical sciences. We can formulate two or more APIs in one dose. Like these two different drugs which I have done is a drug anti-TB and the other is antioxidant, and I have combined the two drugs in a single crystalline form which can have the improved stability. We can also mask the different functional group by hydrogen bonding. Like in this case, the hydrazide group is masked by hydrogen bonding which can easily be broken in the solution form, and we can have improved physicochemical properties of an API while retaining its molecular structure. That is very important modification, that we didn't change the actual structure of the drug and we make a new crystalline form having another component, and they can have the improved bioavailability of an API. The most important thing to synthesize the pharmaceutical co-crystal is to devise the supramolecular synthones, because this depends on hydrogen bonding, and hydrogen bonding can be formed by different supramolecular synthones. These are the structural units based on complementary functional groups and are assembled by non-synthetic procedures involving intermolecular interactions. Like here I have formed the different synthones which can be identified, which can be formed between the isoniazid and the hydroxy derivatives of benzoic acid and cinnamic acid. These are the proposed homo and heterosynthones of these compounds. Here, the synthon C and synthon D are very important, which we found that these are the complementary, where the hydrogen bond is formed between the carboxylic acid hydrogen, and in case of C, the hydrogen bond formed between the hydrogen of hydroxyl group and the nitrogen of the pyridine ring. During our study, we synthesized many co-crystals of isoniazid. And why we selected the isoniazid is that it is highly active against Mycobacterium tuberculosis and being used now against TB. It is a primary constituent of triple therapy to treat tuberculosis, and it is susceptible to hydrolysis and oxidation. So we tried to uh, address this problem, and it can easily form the pharmaceutical co-crystal because of the hidden synthons in isoniazid. As anti-TB drugs cause pulmonary inflammation, so the patient need antioxidants. Also, the fixed dose combination of isoniazid with rifampicin degrades due to its hydrazide group. So to mitigate these problems, we selected the hydroxybenzoic acids and isoniamic acids, which are good antioxidants and are ideal partner with isoniazid to form the FDC. So we tried pharmaceutical co-crystals of isoniazid with these compounds. We synthesized the pharmaceutical co-crystals of isoniazid by two methods. That one is solution method and grinding method. The characterization of these co-crystals was a two-stage step. In the first preliminary identification, we used IR spectroscopy, DSC thermal analysis, and PXRD. And the final characterization of co-crystal was done by single crystal XRD, cast-up studies, and the solid-state NMR studies. These are the IR spectra of co-crystals and suggest the co-crystallization. DSC thermograms also suggest the co-crystallization because their spectra were different from the original compounds. Comparison of experimental and calculated PXRD results also predicted the formation of pharmaceutical co-crystals. Finally, single crystal XRD revealed the structures of pharmaceutical co-crystals, and here as you can see that we can identify the hydrogen bondings present in the compounds in the coformers, which leads to the pharmaceutical co-crystal. During these supramolecular synthon studies, we identified a very important aspect that some compounds lead to the synthon D and some synthon C. So synthon D and C played the complementary role. Solid state NMR finds very important role in the characterization of pharmaceutical co-crystals because the solution NMR is not possible for the co-crystal. As you can see, the hydrogen bonding breaks in the solution form. So this paves the way to characterize the solids which do not form the good crystal for single crystal XRD. So they replace the results of single crystal XRD. So we can use the pharmaceutical solid state NMR for initial identification of screening and identification of polymorphs and identifications of different phases of solids. And cast-up calculations were also performed to validate the experimental proton and carbon-13 NMR results and also to identify the prediction of chemical shifts. Here you can see the result of a co-crystal that was formed between the isoniazid and 4-hydroxycinnamic acid. When we compared the experimental carbon-13 NMR spectra, we found that the chemical shifts of carbon ranges between 159 to 161 ppm for the compounds having synthon C and varies between 164 to 172 ppm for synthon D. After the successful characterization of co-crystals, we determined the different physicochemical properties, and the studies showed that these co-crystals can replace isoniazid in its different fixed dose combinations. Antioxidant studies also showed the promising results and backed our idea to replace isoniazid in different FADCs by these synthesized co-crystals. Our research revealed the successful co-crystallization and characterization and also the applications of solid-state NMR to distinguish between key synthons. Here are some clicks of my solid-state NMR group at Durham University, UK, where most of the experimental work was performed. I am very thankful to all of you and the committee who gave me the opportunity to have a talk with you, and I also want to say thanks to my professors Dr. Uzma Yunus at Allama Iqbal Open University and Dr. Paul Hodgkinson at Durham University, UK.